In this video, we're going to run through kind of like what I would do at this point, you know, in like a grow project like this. Going to grab some different fruits, grab some swabs, grab some tissue samples, and plate all that stuff out. This is where it starts getting interesting, right? So we can start talking about some of these expressions going on in the tub. So I would say on F1.2, it really has this jellyfish thing going on pretty consistently throughout the tub. Wavy margin, almost looks like nutcracker, the way the pileus is like bumpy and somewhat translucent. So I thought that was interesting. I think I might try and do something with that. And I imagine it'll probably not be that difficult because, like I say, that's pretty consistent throughout the tub. There's some of them that aren't quite the same, but generally most of them are doing the same thing. So that's one thing we could look at doing. So F1.3, I think, really has this like kind of the peach look, the Momo look thing from the original images. So really like a really round sort of stipe thing going on, like a ball. These are small, but could work on the size through cloning and stuff like that. But I think that's the kind of the Momo pheno, or whatever you want to call it, that expression. And then there was some in here doing the like wavy margin thing as well, but they don't have that like really bumpy like the nutcracker type thing going on. But I think this is something to try. So I think that might be like the, you know, kind of the like original pheno, or whatever you want to call it, the original expression that I saw the picture of that I wanted to grow, that then not the LC, but the culture produced really weird, all this weird shit. So anyhow, F1.4, similar to, I would say, F1.2, a little bit of different expressions in here, but pretty much the jellyfish sort of thing, tightly wavy margin. This fruit right there, kind of snuggled in behind them, has that like nutcracker cap texture going on. There's also this little short one on the left that has almost like a clockworkish sort of wave to the margin, really tight, and I think probably the bulbous round stipe thing that like the OG expression. So yeah, more of that jellyfish sort of thing going on. But yeah, so those are my thoughts, kind of. You can see like these these few in the middle are kind of maybe gonna do that short squatty thing, bulbous thing, and then getting more of these jellyfish sort of guys out there. So anyway, yeah, interested to hear opinions from the crew here. We're working this shit together, guys, so let me know what you think. Told you my opinions and shit, but anyway. All right, here we go. It's Momo Grow Along. I want to show you. Took a couple days longer than I expected to get into these tubs, and I'm actually kind of glad that I waited. I was thinking of waiting to let these mature, especially these big blobby ones, or big round robust ones. So I'm gonna grab those and probably work with at least one of those here. And then this tub looks amazing. Like, these fruits are so great. This looks exactly like the desired, like, Momo the Momo sort of peach rounded stipe, you know, hand fruit sort of thing. I love it. It's great. So we're going to grab those for sure. And then this is the one that was the more jellyfish tub. A couple other big chonkers coming in here. So I'm wondering if that's maybe what we saw in that initial picture. Oops. I just banged into my light. I wonder if that's what we saw in that initial picture, was something like a later flush out of these. Because, yeah, we're seeing that like in all three of these tubs, kind of. So anyways, gonna grab a few of these fruits and we'll get moving on this. Pretty happy with this this look though. And by the way, these are the F1.2, F1.3, and F1.4. So 3 different F1 culture selections. Sorry, I banged into that light again. So, I'm gonna just put the lids on these 2. And I'm gonna start working with these this tub here. So, I would, in this case, since we're using different cultures, tracking all that kind of stuff, I would just keep track of, you know, what culture these specific fruits are coming off of. So F1.4. So I'll make note of that stuff like on my plates and in my spreadsheet to like keep track of the info. And what I've been doing is using these stainless trays for harvesting and also for taking photos and stuff. The mushrooms look, I don't know, I think they look pretty cool on the stainless tray. It's like a mirrored, almost a mirrored surface kind of thing. So, been enjoying the way those have been coming out. So, I think I'm going to select 2 of these. This big ass one, which I'm assuming if I gave more time would probably flip its cap up and turn into like a wavy scalloped margin thing like this. Oh, this thing is dense. It would be cool to weigh these as well. You know, really connected to that substrate. Okay. Use a little thumb power. Push it from the other direction a little bit. Trying to do this without fucking up the gills and like getting substrate on them, anything like that. Trying to keep this clean, basically. These gills. There we go. Wow, cool fruit. Super cool. Like, it's already bruising heavily. Definitely going to send these away to Mr. Jordan Jacobs to take a peek at, do some HPLC. Whoa, half of the substrate just like bent in half and wanted to come with this huge thing. All right, I'm going to do a twist on it instead of a pull sort of deal. Wow, these are awesome. So cool. Like, look at that thing. So sweet. I'm in love. So that was what I was expecting and kind of hoping for on this my first run of it. You know, it's kind of like the picture that I saw was these rounded stipes, big ball stipe. Cap, I don't recall exactly at the moment exactly what it looked like, but I know it was something similar to this. And then I don't know, there's other stuff in here that could be pursued, obviously. I mean, you see how kind of unstable Shakti is? Just mention that as a reminder that this is a Shakti isolation project. Shakti is an albino Malabar. And originally, Chris Aaron was the one that was, I guess this genetic came from, which I was unaware of, but, you know, pop the lid, or dub top back on this tub, get it out of the way, and split my glove, so I'm gonna also change into a fresh glove. The point I was kind of getting to with Shakti, though, is just it seems to be quite unstable. It produces all sorts of varied expressions, and I personally, I have a couple that I feel like I've kind of isolated or whatever, like the Blue Brain was one of them, and there was another one we put out called Atma that was a bit smaller thing. It's a pretty unstable genetic. It produces all sorts of varied expressions. So I typically like to get this sort of substrate off of these fruits when I go to work with them. But honestly, I kind of think in this particular case, I might do more harm than good by attempting that. So what I'm going to do is just swab these gills. I'm going to leave, I can't get my positioning right here. I'm going to leave some of them intact so I can do a quick bit of microscopy just to keep the spores. I know they're going to be translucent, but I just want to see the kind of maturity level of them on these fruits. These fruits aren't quite as far along like those other ones, like the jellyfish sort of things. The first things that came up in here, those are all turning blue and all this kind of stuff. So I'd imagine the gills, or excuse me, the maturity is totally done with those. But clean hand for swabbing, dirty hand for holding fruit. Basically, if I was doing this project just myself, I wouldn't be making nearly as many swabs. But I'm going to be sending these out for the Grow Along group. So there's that. Yeah, I mean, normally I might make a few sets, 4 or 5 or something at most. And that would be if I wanted to even share it with some friends or something. If it was just me doing my own thing, I might only make a few, a handful of them. And I'm doing that now because I used to make tons, you know what I mean? And honestly, a lot of them just they just go to waste eventually, if you're not doing each project along with it or sending them out, sharing it, and all that kind of stuff, which I do share plenty of projects, but not every single thing I've made, obviously, so plenty of things that I haven't shared. I almost touched the swab head of that. If you ever even hit the swab head, just throw it out or whatever, toss it and just get a new one, because it's not really worth the question of if it's contaminated or not from your accidental touch. One thing also I guess I could mention here is with caps like this that are real tight down, swabbing up in here, you can't really do that, obviously. But the thing I would do, I'm just going to quickly finish swabbing these gills that are out here, and I'm going to leave that little patch right there to cut some gill samples out of to take to the microscope later in the video. So yeah, what I might do in this kind of situation would be to take a scalpel, or sometimes you can just twist them. This fruit is rock solid. It's super firm, so I know if I twist this, it's just going to probably snap into pieces. But a lot of times you can just kind of give them a firm twisting on the cap, and it will release, and then you expose all those gills. to work with. But in any case, I'm going to try to cut this one free right here, right below where it meets the stipe, because I know that trying to tear that off myself would have wrecked it. And while I'm at it, I'm just going to slice these gills free and put them on a microscope slide to take a peek at a little bit later. Slide right here. Now do your best to really only have like one gill laying flat. I mean, you could have multiple gills, but you want to have just one layer thick, essentially, so you can get a microscope slide on it, and then kind of make your crush mount or whatever. Or you can just do, I'll show you the quick way that I do it a lot of times in the lab here, just really quick while I'm working. If I want to check spore maturity or take a peek at like a low spore variety just to see how much spores are there. Or for instance, the other day I was working with the gold spore tamponensis, and I just wanted to throw some on a slide to take a look at it, to see what was going on there. It was cool to look at, by the way. That was really interesting. It was actually like a variegated sort of thing. So there was some purple and then some yellow gold spores. Really interesting. Cool mutation. So in any case, here I've got, try and get this off the tray. Just to see that, yeah, a few, I know it's focusing behind it, but you can still sort of see a few gill fragments on there. And there's a couple here that are more than one thick, like what I was saying. So I'm actually gonna cut that off. I keep straight razors. I just rearranged my lab, trying to figure out where the hell I put them. I keep straight razors in the lab for doing like microscopy slide prep. Here we go. Sorry, I just put all of my supplies on this little thing over here. I guess I can show you in a second since it isn't in the lab. Probably want to see what's going on in the lab. I've been doing a lot of rearranging and cleaning and stuff lately, just trying to get things more optimized. But anyways, just a regular straight razor works really great for this. And you can cut it into very thin pieces this way, and then use like a scalpel or a very fine tweezer to try and sort of arrange them into a nice like field to view them. So I know this is riveting for anybody that doesn't do microscopy. This is probably like the most boring shit, but for anyone that is trying to learn microscopy, hopefully this is a semi-helpful thing for you guys. Yeah, that's just one thick. It's just starting to curl a little bit because it's getting dried out from the air current of the flow hood. I'm just going to discard this piece. No, it's fine. I'll just put it on the side here. We can take a peek at it to see what's going on with it. It'll probably be kind of dehydrated. And then this big old chunk. Yeah, nice. That's just one as well. Just clip off this edge because it's kind of a little thicker, sticking up. You want to try and get these samples like thin if you can. That's what the razor blade is really nice for that. And then, like I say, just positioning here. I want to get them down here into the same kind of, just the same area, essentially. Okay, that'll do. You could throw this straight onto the scope and just look at it as it is, which is what I do a lot of times when I'm just doing like a really quick thing and just want to quickly peek at a feature. But you can take, to make a little bit nicer mount, take your scope slides. This thing's a little dusty, so I want to wipe it off. And make a little mount. And I don't have any KOH, which is what typically you would want to do. Is have a fully dehydrated specimen and then rehydrate it with a little bit of KOH, and then do this scope, microscope slide application, whatever you want to call it. And this is kind of messy, so I'm just using the ISO alcohol that I have sitting next to me. And a lot of times I'll just use my shirt to try and kind of wipe it, get any dust and debris off of there, because it's going to obviously kind of impact your view when you're trying to look at the specimens. That's not perfect, but better than it was when I started. So I'll just leave this right here for a second. How about I leave it right here for a second? And what I normally do is just add a little tiny bit of distilled water, which I have in this bag here. Some syringes. And I use these for a number of different things in the lab. I guess this would be its whole own video or whatever, potentially, just talking about different ways to use a syringe of distilled water. Maybe not a full video, but in any case, this is one of the things I would do with it. I sterilize those with my agar. So it's just like an XLSA grow bag and some 10 ml syringes that have water in them. And you don't want to fill the thing like all the way full, so I leave a bit of room, because otherwise, like, you know, it boils, because it's getting so hot and everything, it just boils out. And you lose a bunch of the water and everything is messy. So anyhow, basically here you just want a teeny little bit of water on your scope slide, and you can put that down between these samples. Like so. It's best to, what I did was use the plunger to get the water up there, and then I just squeezed here to get a little one drop to come out. Typically, if you use the plunger, it's going to shoot way more water out than what you want. So we have our specimens on the left with the drop of water. The ones on the right are the thicker ones that I sliced off with the razor blade. And then we have our microscope slide cover. You just want to kind of put it on here and then drop it down like at an angle on here, and then just bring it down at an angle. And this is to help get rid of air. So you push it down from the back forward. And that's like a super simple crush mount. It's like not going to be a really great way to view this thing, but it'll give us like what we need to see. So Functional, at least. Not the greatest, but anyways, that's one of the ways I'll just do a quick sample prep for the microscope, and then set this off to the side so I can finish working with the fruits, and then throw this on this microscope later. Mark this as Momo. What did I say this was? F1.4? F1.4. So that way you always know what it is. Just like labeling a plate or something. Alright, so anyways, let's get back to action here on the fruits themselves. I was in the middle of slicing that cap off, and I detoured to make that whole little slide prep thing, so apologies. I do apparently have ADHD according to these doctors. So, there you go. I don't know. Lay this over here in the flow so I can use it later for something else. And we get this cap off of here. Let's go. Finish working with these things before this video ends up being 6 hours long. I'm trying to separate the cap here from this stem, large stem, that's what it is, with a gigantic stipe below it, and do so without breaking all these gills and messing them all up. So looks like we got it pretty good. Yeah, nice. So that's what I'm talking about, is all those gills that were obscured by the stipe, now you can get at. So a lot of stuff like ape, for instance, or these things that have tight caps that hug up against the stipe. That's a way that you can get at the gills. I'm also doing these swabs a little rougher than, how I'd even say it. I'm picking up some gill tissue, almost like intentionally, because they are going to go into the grow lung, so we'll get some spore stuff, probably some even tissue regeneration also. All right. Pretty cool bluing reaction. We got our blue peach. All right, so I'm going to keep these separated. What I'll probably do is call that like F1.4A, another one F1.4B or something along those lines, just to keep things somewhat separated. I can see the gills. It has like a little tiny neck. I don't know if this will come through on the video. Got a little neck up in there. So this cap is pushed up far enough that I can get up in there. I didn't think I was going to be able to, but yeah. All right, so this will be fine. Get these swabs done, and then we'll take the tissue samples. This thing's really turning dark. It's almost like the color's gonna come through or not because of the lighting, but it's almost turning like black. I don't even know how many more of these things I really need to work with. I'm pretty happy with this. Alright. little bit of ISO to the hands. Use a soapy spritz bottle as well would be another option to use in the lab instead of ISO. I'm going to let these swabs dry a little bit and just remember that this was that fruit and that thing is from that fruit. Let's see. Move this stuff back a little bit, get this cup out of my way. And we will grab I'll just throw these on a couple 60 ml plates. One of the things that I like about the 60s is I feel like you can pour like 1,000,000 of them out of one 500 milliliter bottle of agar for one. And then on top of that, I don't know, they feel like a good thing to use for like clones and that sort of stuff. These ones are a little messed up actually, but it doesn't matter. For this purpose, it'll work just fine. But I'm not sure what even caused this to happen exactly, but there's like, they split. The agar puck split. It's kind of, I don't know what caused that. But anyways, because these fruits are rock solid still, I am going to use this razor blade to... that one I might be able to just pull apart. Let me see this other one. Sometimes you could pull these things apart, but if it's really, really firm, it's better just to use a blade and only cut a little bit and then take your sample away from where you made that slit. I feel like that's better. I know that people always say don't cut into the mushroom, but honestly, you're going to do more harm than good by manhandling a thing as opposed to just contaminating a small part of the flesh and then just take your sample from a different area. It seems like a pretty simple solution, but I made that slit and then I'm using my thumbs, pushing down and in to try and get this to break apart. There we go. Pretty awesome. So inside of here should be sterile. Where I sliced it, not so much, but that's why we're exposing the rest of this. So I'm just going to grab 2 samples out of here. The first one I'm going to take from right here. Cut this piece free. There we go. The other piece I'll take from, let's say right here. Pretty nice, nice dense, solid fruits. Yeah, really cool. Okay, so that would have been fruit number 2. And just because, you know, things you can lose track of this kind of stuff pretty fast. I'm just going to mark these things really quick with a little tape thingy, so I don't mess it up. So I'll just call it fruit B and A for the moment. Give myself a little reminder on these swabs. And I'm also going to write the same thing on those on the plates. For my plates, I use always use an alcohol-resistant marker. So Sharpie is not used on my plates at all. So yeah, that was B fruit. I used to use Sharpie on my plates and stuff, and you know, you wash away a bunch of important information as you spritz your plates down and try to get things clean. Got a little few pieces of parafilm pre-sliced down here. Thought I had two in my hand. Maybe I dropped one, or it didn't come out of the bag. I wasn't paying that close attention. Anyway, can typically wrap 260s with one strip of parafilm anyway, so get it around twice, and then we'll get our sample from fruit A. Again, I might be able to just just tear that apart, but it does feel kind of like sketch. Like I would do more damage than good. One other thing you could do would be to take this and carefully open that, and you've got clean tissue in there. So why don't we grab one from here? And one from inside this thing, this stipe structure. These are fucking awesome. I love this. I love these fruits. These are really great. This one is not quite 100% solid, like that other one was. This one's a little fluffier inside, actually. So that's kind of like, this one is not as dense for that interior section. Actually, really similar. Yeah, it is actually a little spongy in there too. So anyways, I'll let those grow out a little bit, and then basically just enough to get some healthy mycelium going, and then subculture them to their own plates. I do it like this because in this case, most likely what I'm working with is going to be clean and everything, but you know, a lot of times you run into like yeast or bacteria and stuff like that when you're working with some different, you know, like if you're working with wild fruits or maybe something that's a bit older, past its prime kind of thing, whatever you want to say. So doing a few different ones, you know, lets you hedge your bets in a way, kind of, and that way you can be pretty certain you'll get some clean mycelium to work with off of your plate. Sorry, I'm looking for some Petries. Here they are. I knew I had some out right in front of my eyes. So I'm just going to grab 2 of these and streak one. Actually, you know what? I'm just going to streak one of them. Which one do I do? If I had a live poll right now, you could choose fruit A or B for Yoshi to streak. That'd be kind of fun, right? I'm sure I do live videos like that, though. Been doing a lot of it live in the lab sessions, like connected with the Discord, and that's been going good. But sometimes you get some, whatever, you know, people want to cause problems. So yeah, I think I'll just do from fruit A. Looking for one that's like the least tissue laden, essentially, for the bottom stuck to the top. For this kind of work, you're wanting spore regeneration, not just clones. If you want clones, you can take tissue clones from the fruits themselves. For me personally, what I'm trying to do here, and I'm going to put this back in here, which I wouldn't normally do if I was selling swabs, but since we're just giving them to each other, that swab will be totally fine to use again. But anyways, what I was trying to say is what I'm looking for here is spore germination as opposed to just tissue regeneration, because you know, if we want to, doing cloning is great, which is kind of a big part of what this project has been started with, is the 8 clones that were distributed to everyone, and then the original LC, also a few spore swabs to the few people that they were available for. But in any case, if we're going to try to like isolate something, traits through spore generations, then that's what we need to do. Doing it through cloning, like if you took spores from these clones and ran them, you're going to get a varied bag of expressions, most likely. So doing multiple grows from spore, advancing that F number forward, would help to potentially stabilize these things. Now, at the beginning of the video, I was going on and on about how unstable Shakti is, and that's true. So actually isolating these traits, we'll see what happens. Sometimes it's sometimes you just kind of have to go with the flow, is what I say, and work with what they give you, because I don't know, you can't necessarily control exactly what comes out the other side. So this would be Momo. F1.4. And what I call these is just like S1. So this is a streak. S1, streak plate. I'll date it. It's the 18th. And then let that grow in, and I'll take selections from that. And then those will become F2. So it would be like, you know, actually this is F1.4A. No, F1.4A streak. Since it came from that one fruit, I want to make sure we keep track of that info. But yeah, so let those grow out and then make selections, and then those would become like Momo F2. What would it be? F2.1, F2.2, blah blah blah, down the line. You can also keep the A letter in there to try and keep track of the designation, like where the plate came from, or where the spores came from initially. Alright, so this would be Momo. What we had before was C2, so these are going to be C3, C3B, and C3A. I was going to use the full labeling system right now that I shared, the Imperial labeling system from Julian. I would put a 29 in front of this, but we all know here what the score is. Alright, so we got Momo C3A, Momo C3B, and dated late. So more than anything, these are kind of temporary plates anyways. This is just to get growth going and then transfer things to, isolated plates of their own. But in any case, that's the tissue samples, the spore streaks. Now I will take care of these spore swabs and put them back in the packages. And I'm going to do this like in sets, A and B, since we want to try and keep things separated. The expressions were pretty similar, really. So sometimes separating things this granularly doesn't even matter, but you never know. Never know what you're going to get out of these things. So sometimes being granular like that is a good idea. Alright, this is going to be a sped up part of the video. Riveting stuff, sealing swabs. Okay. Move the camera like that. Alright, catch you in a minute. Now we have our swabs separated and labeled. Set these aside.